back up real quick, and I want to start at the very beginning, because they say that's a very good place to start. And so I want to start with target identification and validation. And Monika, I'm interested in your perspective on perhaps what are some of the challenges around target ID and validation in ADC specifically, and what tech advances like computational and AI are potentially contributing to that cause right now.
I think target identification is essentially the first step which we have, when we are looking at ADC development, or for that matter, any drug discovery. And, we are actually aiming to identify, let's say, molecules, usually proteins, that you could alter a disease state if their activity was modulated. So of the approximately, I believe, 20,000 proteins in the human genome, only about 3,000 have been identified as potential therapeutic targets. So you can understand that, there is still a lot of potential targets which we can identify, and also repurpose targets which we have already identified. And I think machine learning algorithms can definitely help to accelerate this process, because they can look at various types of data, including, let's say, gene expression profiles, protein-protein interaction, genomic data, proteomic data, and to identify potential targets that are likely to be involved in the disease pathway. So I believe this can be done at a much more accelerated pace by using machine learning algorithms. Not only for target identification, I also feel that as far as molecule optimization is concerned, let's say the optimizing compounds is concerned, or estimating their bioactivities is concerned, or looking at protein-drug interaction is concerned, AI can be used to virtually screen and optimize compounds. And one way of doing this is to look through the development of predictive models that can, identify compounds with a high probability of binding to a target protein. And these models can be trained using various types of data, whether it is protein ligand complexes, or structure information, or molecular descriptors. I think we've recently had some success as far as protein structure prediction is concerned with AlphaFold breakthrough, or we've recently also had foundational models when, they have provided an open source for protein structure. And with foundational models, we know that a lot of time and cost is also saved, because you already have a base to work on or to carry out your computational tasks as far as biologics is concerned. So I think that has really helped in looking at target identification as far as AI or tech is concerned.
Yeah, thank you, Monika. Léo and Philipp, I'm wondering if either of you have any follow-up there, if you can speak at all to perhaps adoption of some of these computational tools at Araris or Debiopharm. I mean, I know there's quite the continuum in terms of companies that are adopting this at a rapid pace versus maybe just dabbling in it and trying some things out. So just curious if either of you would like to follow on to Monika's comments there.
Yeah, maybe I can start. So
Go ahead.
So Léo, please go ahead. Yeah, okay, so thanks. Yeah, I mean, target identification, it's a very interesting topic. And I think we in the field learn every day more about the targets, and also the importance of them, of course. Because when you look, for example, I mean, a recent example of the importance of whether it's a good target or bad target, it's TROP2. I mean, it has been around for years. People think, and I think now when you learn about the behavior of the TROP2 in clinical development, whether it's a good target for example, for the lung patients, and I mean, it was AstraZeneca that presented, I think, last year, some data how this target is distributed within the cell, within the tumor cell. I think it's quite interesting to learn that potentially the target may have, like how it internalizes, how it's distributed within the cells. I think it will become a critical aspect of whether it's a good target or a bad target. And yeah, it may also be different in different indications. So I think the target can be really different in different indication, and that's why also learning more about the target and maybe use AI to learn more about it. I think that will be help to further develop exciting ADCs. So.
Thank you,
Yeah,
Philip.
to add to that, since Philip mentioned about QCS, I think one of the big advancements which we saw last year in terms of AI, or, I would say AI-driven pathology or AI-driven computational pathology, was in the development of ADCs. When we looked at the QCS algorithm and we found out that computational pathology or computational techniques were able to not only identify the intensity of expression, but also identify the pattern of expression, as Philip mentioned, at the single cell level, also to identify the dynamic range of expression. And importantly, it helped to, let's say, classify patients depending upon the spatial bystander effects. So artificial intelligence and computational algorithms are now helping us to not only look at the tumor cells, but also looking at the tumor microenvironment when we are looking at spatially bystander effects. And this is especially very important with ADCs because we all know that ADCs, we typically feel that, okay, they have the potential to deliver a toxic agent or a potentially cancer-killing agent to the tumor cells, but they also have a very narrow therapeutic index. So it is very important to identify the correct patients and to make sure that the patients are stratified in a way they are reacting to that particular biomarker. I think this is where not only optimizing the patients, optimizing the clinical trial design, and in a way also expanding the patient pool becomes important, and it becomes more helpful in terms of computational and AI-driven pathology.
Excellent. Thank you, Monika. Leo, I think you had something to add there?
Yeah, I think AI has also two other places where AI and modeling can play a role. I think there's a lot of modeling you can do in terms of DMPK of your ADCs. I think about the work of Greg Thurber at University of Michigan, who can really predict the efficacy of your ADCs with all your in vitro parameters and run all sorts of modeling, to monitor tumor penetration and cell internalization, trafficking, and so on, and eventually even try to predict some efficacy of your ADC in patients. So using your in vitro data and your initial PK data and running all these very complex mathematics with computational tool allows you to predict your ADC efficacy. So that's these types of tool are emerging, and my dream is that one day such tool will be like available to really to ADC scientists as platform, as a software to use. But the things are coming, in that field. And another area maybe is also, simple vitro work is sometimes guided by AI. Before you were just doing cell titer glow to measure cytotoxicity. Now you have these equipments that can detect cell death, in your in vitro assays directly, So and then you have AI there that can compile your cytotoxicity result in a much more precise way, and you can look independently at cytotoxicity or at just cytostatic effect and so on. So these equipments are used here to get a lot of information on your ADCs, and then can sometimes make a difference with peculiar classes of payload who do not always display cytotoxicity in vitro, but then when you start looking at these closer parameters, particularly in terms of cytostat with detected by those tools, you see a difference, and you can refine your ADC ranking maybe in a better way.
Yeah, I'd like to sort of weave this computational AI question throughout our discussion today as we talk about some of the challenges in process development. But I'm going to ask one more question, and then I'm going to get to a few audience questions because they started rolling in the minute that we started talking. And Monika, I'm going to direct this one toward you. Philip Leo just spoke of the dream right? The software application in AI. What stands in the way, Monika, from your perspective? What stands in the way of sort of larger scale adoption of these computational tools?
I think, to be honest, at the moment, there is lack of adoption of, whenever we talk of AI tool, there has to be, generalizability, wherein we have accepted validation criteria for clinical use. We also need to have standards and validation and regulatory standards for AI algorithm across laboratories, and especially as a tissue pathologist, because I deal with images. So I would also say that, the generalizability across and acceptance of this technology using different scanners, using different image management platforms, including different monitors. I think there has to be more a generalized standard across this so that there is seamless integration. So this would also need robust IT infrastructure, which can support comprehensive, AI-driven analysis. We also need, I would say, accepted validation criteria for clinical use, which has regulatory approval. Also, one more important aspect would be explainable AI, as we call, we do not want non-transparent or black box algorithms. We want multidisciplinary teams working from the time we talk about AI algorithm development, whether in terms of tissue or whether in terms of anything else. And also we need imaging standards for adopting this across laboratories, and also to have this robustness of these AI algorithms, whether in one lab or in another lab or in another setting. I think there should be validation criteria for determining the robustness of these algorithms across different laboratories and across different settings.
Yeah.
And obviously, we need a very good IT infrastructure for this. So.